Excellent. Welcome, everyone. Welcome to this very first session of our 6-month Biocontrol Technical Workshop series under the ASEAN Fall Armyworm Action Plan. My name is Alison, and I'll be the moderator for today's session. I'm joined by two experts from CABI, Roger and Marc, and Marc will be doing much of the presentation today. We also have our technical expert for our webinar platform, Pranav, helping us out. So if you have any problems, you can message him in the chat box. And we have Graham Dixie, Executive Director of Grow Asia, joining us as well. Just to reiterate, I showed a few people this before, but we do have a really big session. We're going to break it up into 3 parts, and we'll have question and answers in between. It will be a bit flexible, so we'll see how we go and see how the session runs and where there's good times to stop. But please really take your opportunity here to ask our expert, Marc Kenis, all the questions that you have on this topic, and we'll try and cover all of them. And if we don't cover some of them, we'll write those down and come back to you. So here is how you will interact with the platform today. You'll write all your Q&As in the Q&A box, please. It's really helpful for us to have questions all in one place. The chat is there for sharing documents, your research, thanking a speaker, sharing a link, highlighting an important point. We really welcome you using that as well. If you can rename yourself, or just make sure that your name is correct, and you can do that by going up to the top More box there, which is under number 2, and you'll just press that and you'll have a little place to rename yourself. So that's most of the sort of highlights for how you interact. I just want to reiterate, this is the first part of the 6-month series. We're really excited to launch this. It's supported by Grow Asia, CABI, IPM Innovation Lab, BioRationale, International Rice Research Institute, IRI, ICRISAT, and the Chinese Academy of Agricultural Sciences. And we'll be bringing on more experts as well as we go along. Anyone is welcome to join this. It's not just for those in Southeast Asia, but if you're further afield, please stay with us, but also we welcome others attending as well. And we really want an interactive 6-month sort of workshop series, and we would really urge you to join the discussions, post your work, share your comments, ask questions at the CABI research portal, and I've provided the link there. And after the session, I'll also email everyone that attended today's session with that link as well. And just to give you an idea of the wealth of topics that we'll be covering over the next 6 months, I mean, this is quite outstanding. I'm sure if after today's session, you'll be hooked and you'll want to come and watch all of them and participate, but you'll find something for everyone there. So please spread the word, because we want lots of people participating, lots of people interacting, and we really want to hear your successes in the field and your failures over the next 6 months so that we can learn from that together. As I said, there's the agenda. We saw that before. I'm just going to launch a poll quickly. Pranav, if you could launch the poll. We've had over 313 people register for this event, which is incredible. We just want to get to know you a little bit more, and I've put in lots of different categories there, so I'm sure you'll find something for you that best describes who you work for. And we'll give people just a little bit of time to answer that. We've got 117 people on so far, so that's pretty exciting, because I know it's pretty busy coming up to Christmas for some people, also the end of the year for other people, and we've had a lot of webinars this year, so it's pleasing to see everyone joining us for this event. 121 people. I'm going to stop it when I get to 90. Oh, there we go. Polling is closed. So we've got about 39% government, and I guess I can just... Can everyone see that? Yep, you can view the poll results. 32, a third research institution students, and 14% private sector and international organisations. Okay, that's good. So what we're going to do now is we're going to get started with all of you, and we're going to move to the next slide. And I'm going to welcome Graham Dixie, Executive Director of Grow Asia, just to say a few words to welcome you to the session. Graham.
Yeah, thanks, Alison. And first of all, it's my really pleasant role to welcome you all on board. It is astonishing to me just to see how the community of practice that Alison and the Regional Action Program has whipped up, with over 300 people registering and well over 100 already attending. It's an extraordinary achievement, and particularly in contrast to see what we're seeing on other kind of webinars. Well, I thank Alison for setting out a 6-month program, 8 different events, ending up in a workshop in June. There's a lot of meat here, and it's an important area, this biocontrol. This is really digging in about how we can control this particular pest without having to recourse to agrochemicals. And this is such an interesting area insofar as what we're hearing is that the market for biocontrol products is growing by a compound annual growth rate of 25%. So it's an important area. It's obviously an area of interest, and it's going to be an important tool in controlling this pest, which we estimate could, if not controlled properly, cause the farmers in our region over $800 million of lost income per year. So back to you, Alison, and over to Roger.
Excellent. Thank you, Graham. Thanks for the introduction. And we couldn't do all that organisation and have all those experts without all our coordinating partners who we really value. And one of those partners from the very start has been CABI, and it's I'm very pleased to welcome Dr. Roger Day to provide a few thoughts.
Thanks, Alison, and thanks, Graham, and hello to everybody again. It's great to be co-hosting this new series of webinars, and we thank Alison and Croatia for all the work they've done to put it together. Biocontrol is a subject very near to the heart of CABI, so that's why we're very pleased to be leading off this first webinar in the series. We always talk a lot about IPM, integrated pest management. Biocontrol is the basis for IPM, really. So this is a critical area in managing fall armyworm. And today's topic, I think, is the basis for all biocontrol investigations, whether it's products, augmentation, classical biocontrol. You need to start all that with knowing what natural enemies you have. And that's what Dr. Marc Kenis is ideally place to talk to us about. He's been doing this for many years, and he's been working on surveys of natural enemies of fall armyworm in Latin America and in Africa. So he's undoubtedly got some very good tips for us all. Thank you, Alison.
Yes, indeed. And it's a pleasure to have you on board, Roger, and the CABI team, and a pleasure today to introduce Dr. Marc Kenis. And I think you've given him already the introduction, so you've done the job for me, Roger. But Marc, it's a pleasure to have someone with so much experience, over 30 years of experience in applied and environmental entomology. Marc is also currently leading the risk analysis and invasion ecology section at CABI in Switzerland. He really is an expert in this field, and it really is a pleasure to have him join us. So Marc, I'm going to give you the floor from now, so you can start when you're ready. If you could you can choose to put your video on or choose to have it off. It's up to you.
Oh, well, yeah, I think it's better to keep it off. Oh, you want to see me first, maybe?
Yeah, that would be nice. Hello.
Okay, hello. For the moment, we still see the presentation, but yes, nice to see you. Good afternoon, everyone, or good morning to those who don't come from Asia. Okay. So I'm going to talk about, I've been asked to talk about field collection and identification of fall armyworm parasitoids and predators in the, just a technical question. Can I switch off on my screen all the messages that come and hide my slide? The chat, can I switch off the chat or not?
Yeah, you should be able to close them, Marc. There should be the arrow pointing down. You click that and you hit close. Okay, because there are messages that come all the time on my screen and they disturb me from the slide. So it's a bit complicated to, I don't know how to do. Sure. On the top left of the message box, there should be a little arrow pointing down. If you click that, another thing will pop up that says close.
Okay. Okay, I don't really see it, but it's okay. I will manage with. I will try not to read them. Okay. So this is so this is a talk about field collection, identification of fall armyworm, parasitoids, and predator. Don't expect me to give a very exact protocol, because I think that this kind of field particularly needs a little bit of flexibilities and need to be adapted to all the different situations. So I really don't advise you to necessarily follow very strictly any protocol which you may find be used elsewhere, but give you the flexibility to adapt it. Next slide, please. Yes. So the plan of the presentation is as follow. I will start as a kind of introduction to talk about parasitism and predation of fall armyworm, and why is it important to know the local natural enemy complex before developing biocontrol projects. I think this is a very important question, because I see too many people who like to study the natural enemies, but at the end they don't know really what to do with these data, except by saying that yes, there is already or there is not yet a biocontrol going on, but nothing else after. And I think it's very important to understand why we do that. And the second, and this will be the most important part of the presentation, is how to assess the natural enemy complex and their impact on fall armyworm population. Impact means parasitism rates and predation rates and so on. And I will finish with a short section on identification of natural enemies. Next slide. Of course, if we study natural enemies, it is because it is directly related to biological control, and I feel I still need to give a definition of what I mean here by biological control very quickly. So biological control is the manipulation of living organisms, which are natural enemies, to control other living organisms, which are pests. So this definition is important, because it means that it is only when you are using living natural enemies that you are doing biocontrol. There are lots of other methods, like for example pheromone trapping and so, which are often included into biological control, but in the strict sense of term, it's not biocontrol at all, even if it can be very well seen as well, and very positively seen in IPM programs. So we really talk here about natural enemies. Next slide, please. And what are the natural enemies of fall armyworm, especially in the area of origin? Well, in the area of origin, the first category, which is the best known natural enemies, are the parasitoids. They are the best known because they are the easiest to study. So parasitoids are wasps or flies which parasitize one single host during their life. They can, and in the Americas, there are over 100 species which have been recorded as natural enemy, as parasitoid of fall armyworm, both in North, Central, and South America. Some are purely egg parasitoids, which means they parasitize the eggs only. Some are egg larval parasitoids, which means that they parasitize eggs and come out and kill the larvae, usually medium instars. And next slide, please. And but the majority are larval parasitoids, and they can attack small larvae, early larval stage, or old larval stage. Most of the parasitoids which attack the old larval stage of fall armyworm are Tachinidae, while usually the wasps attack earlier instars. And some of these larval parasitoids also emerge from the pupae rather than from the larva. Next slide. And of course, we should not forget the pupal parasitoids. they are much less known, especially because it's much more complicated to sample pupae in the soil or in the litter than the larvae and the eggs on the foliage, but they should also be better considered. Next slide. And then the other category of fall armyworm natural enemies are predators, which feed on more than one prey in their life, otherwise they would be parasitoids. So the predators, most predators are insects, or at least arthropods. They can be earwigs, ants, bugs, beetles, spiders, etc., etc. But we should not forget as well the vertebrate predators, for example, the birds, the insectivorous mammals. Frogs are also sometimes important predators of fall armyworms, etc., etc. They are less well known for reasons I will mention later. Next slide. And then I just want to quickly mention that the third category of natural enemies are the pathogens. They may play a very important role in the population dynamics of the pest, virus, bacteria, fungi, microsporidia, and so on, and also entomopathogenic nematodes, which are not directly pathogens, but they carry a pathogens that kill the host. So these ones will not be the topic of the talk here, because there will be other sessions later on the pathogens which are used for biopesticides, and so I'm not going to talk about the pathogens. But parasitoids and predators need to be studied in the area of introduction in Asia, in Africa, and so on, for different reasons. I guess you probably know that there are 3 different categories of biological control methods, and for all these categories, it's important to know the natural enemy complex in the region of introduction. The first category, just to remind you, the first category is the biological control by introduction, or classical biological control, which is the introduction, for example, for fall armyworm would be the introduction of a parasitoid or a predator from Latin America to introduce into Asia or Africa. That is for permanent establishment and control. This is classical biocontrol. The second category of biocontrol is biological control by augmentation, which is the augmentation of the density of natural enemies by regular releases. The release can be inoculative, which means that you inoculate at the beginning of the season or the year a small number of biocontrol agent that will reproduce, but you need to do that year after year or season after season. Or inundative, which means mass releases for a single and immediate control, which in this case is quite similar to the use of pesticide. In the case of fall armyworm parasitoids or predators, this is, for example, the case when you release trichogramma or telenomus to control, to parasitize the eggs in an infested field. The third category is biological control by conservation. This is usually much less known, but this is all the methods which favor the efficiency of natural enemies which are already present in the system, but for the moment are not very efficient. This is, for example, in this case, if you want to do intercropping, or if you want to reduce pesticide and so on, you will enhance the natural enemy complex. And for all these 3 categories, there are good reasons for studying the natural enemy complex in the area of introduction. Even for when you are strictly only doing biological control by introduction, which means just introducing from the Americas a natural enemy for permanent establishment and control, you still need to study the natural enemy complex in the area of introduction for various reasons. Next slide. The next slide, yeah. And the first reason is that the candidate natural enemy may already be in the region of introduction. And a good example with fall armyworm was Telenomus remus in Africa. When fall armyworm arrived in Africa, there were quite a few people or organizations who were thinking of introducing Telenomus remus, which is an egg parasitoid occurring also in the Americas, to Africa. But thanks to surveys, in Africa, we found out, and many of us found out in different countries, that Telenomus remus was already present in Africa. God knows how it arrived there, but it was already present. So there was no need to introduce this. But we would not have known, of course, without a proper survey of egg parasitism in the different countries. Next slide. And the second reason is that it is usually, when you do classical biocontrol by introduction, it is always better to fill an empty ecological niche than introducing a species that will compete with a local natural enemy. And a good example with that is with braconids of the genus Kenis, these egg larval parasitoids. So in the Americas, Kenis insularis is probably the most abundant, the most frequently cited parasitoid of fall armyworm. But also when research started in Africa and then later in Asia, we also found out that other Kenis species were also among the most important parasitoids of the fall armyworm in the introduction region. And this needs to be taken into account when you want to select a parasitoid. It may be better to select a native parasitoid in America for introduction to Asia and Africa, which does not compete directly with very similar species. Next slide. And then finally, post-release studies require baseline knowledge of parasitism and predation in the area of introduction. What does it mean? It means that if you want to introduce a parasitoid into a new region, so once you have done that, you need to know the impact of this parasitoid, including on the natural enemy complex in the region of introduction. And if you don't have these baseline studies to know what happened before the introduction, you cannot do that. Next slide. So the second category of biological control method is biocontrol by augmentation. So the regular releases of natural enemies. In the case of fall armyworm, it can be egg parasitoids or predators, bugs, for example, and so on. Well, then in this case, the reason for studying the natural enemy complex in the area of introduction are as follows. First, of course, you need to identify potential candidates for mass production and regular releases. So that makes sense. And also that it's much easier to obtain permits for producing and releasing biocontrol agents which are already present than by introducing natural enemies for augmentative releases which are not yet in the region. And it's also environmentally safer. There is no environmental risk or much lower environmental risk to use for this kind of releases natural enemies which are already present than those which need to be imported and go into all the procedures. No, please, can you go up? Can you go back? Sorry. Yes. And a good example is the story of Trichogramma. Trichogramma is one of the two egg parasitoid genera which is used against fall armyworm, the other one being Telenomus. And in the Americas, they are using Trichogramma pretiosum, which is an American species with some success, medium success. Trichogramma pretiosum does not often occur naturally. But when the fall armyworm was introduced in the different countries in Africa and Asia, there has been a lot of pressure to introduce also Trichogramma pretiosum, because Trichogramma are very easy to produce, to mass produce, and they wanted a Trichogramma which is known to work more or less against fall armyworm. On the other hand, when surveys were made in Africa and Asia, there were other Trichogramma found, like for example Trichogramma chelonis, was found in several African countries. It's also found to be abundant on fall armyworm in Asia. So, and actually the rate of parasitism of Trichogramma chelonis is much more important in the invaded region than the rate of parasitism of Trichogramma pretiosum in the area of origin. So why would you introduce Trichogramma pretiosum if you already have Trichogramma chelonis, which occurs naturally on fall armyworm? But of course, you would not have known that if you had not done the surveys of egg parasitoids in Asia and Africa. Next slide, please. Yes, and then of course, exactly for the same reason as classical biological control, even for augmentative biocontrol, we need baseline studies on parasitism and predation before releases to see whether there is any kind of impact on the natural enemies, especially on egg parasitism. If you want to release egg parasitoids, mass release egg parasitoids, you already need to know what is the natural rate of egg parasitism in the region, of course, to assess also the success and the potential impact on the biocontrol agents. Next slide, please. Okay, then biological control by conservation, the third category, which are all the methods favoring the efficiency of natural enemies already present in the system. Of course, there the reasons for studying the natural enemy complex become immediate. You cannot favor the efficiency of natural enemies already present in the system if you don't know these natural enemies already present in the system. And only a very good knowledge, not only a list of natural enemies, but also a list of the factors which enhance or suppress them will allow the development of conservation biocontrol strategies. For example, you can enhance them by using intercropping rather than monoculture, or flower bands and so on, or for example also chemical pesticides. You can also moderate chemical pesticides, for example, by changing to biopesticides, or by lowering the amount of spray, or by changing the timing to enhance the natural enemies. But for this, you need to know exactly which natural enemies occur when and to which level. There are also other ways of enhancing natural enemies. For example, in Africa, in some countries, they are using fish soup and sugar to attract ants and things like this. But all these methods need a very good knowledge of the natural enemy complex in the area of origin. In the area of introduction. And with this, I think I finished the first part. Alison, do you want to take the questions now?
Hi, Marc. Can you hear me?
Yes.
Yes, what I think we'll do is we're just waiting for some questions to come in. I think that's a very good introduction, and that's exactly what we need to start. I think there'll be lots of questions coming up when we get into the more technical, how do you do it on the ground? Just a quick question to you. Is CABI doing any work at the moment within Southeast Asia or neighboring countries on any of these three approaches, classical, augmentation, or conservation?
Yes, of course, we have a center in Malaysia, and our China center also works on this in this approach, but not necessarily yet much on fall armyworm because it just arrived. Yeah. But on many other pests, yes.
And is there? I thought there was something potentially around fall armyworm from a classical some work there. Is that true, or did I hear wrong?
Sorry, I didn't not understand.
Sorry,
the question.
was there some CABI work, I think, around a classical introduction of a particular
yes,
predator?
not yet in Southeast Asia. For the moment, we are working for Africa and with Pakistan.
Okay, Pakistan. Yes, no, that's what I'd
Pakistan,
heard.
Yes,
and how long does that take? So what stage is that up to?
Well, it takes some time because classical biocontrol it takes a long time to develop because you need all the risk assessments before before being allowed to release a natural enemy. So there are lots of different steps. You first need to assess which natural enemy is among the most efficient in the area of origin. Then you have to check whether these parasitoids or predators are safe for introduction.
Yeah.
This needs a lot of work in quarantine, which is much more complicated than outdoors. So it takes usually several years. It can take 5 or 10 years before the introduction
5 to
of
10
a
years.
natural enemy. Yeah.
Okay, excellent. I've got a couple of questions here. Yes, so
Alison,
Roger.
this is Roger. I was just going to chip in and say, I mean, several years' work has already been done, and we have imported a natural enemy, a parasitoid of fall armyworm, to our quarantine station in Pakistan. So
yeah.
the first candidate is in Asia, but under quarantine, and as Marc says, it has to undergo a number of safety tests, if you like, host specificity testing before any release could be considered.
Yeah. No, that's good. Thanks, Roger. That's exactly what I was referring to, what I'd heard about. So that's very interesting. I've got a couple of questions here, Marc, and then we'll move on to the next part of your session. Is there any hyperparasitoids for those specific parasitoids mentioned? Is this from Cyril?
Yes, there are. There are some. We haven't found many so far in the region of introduction, but in the area of origin, there are hyperparasitoids which are known. Yes, I mean hyperparasitoids are not that prevalent in fall armyworm if you compare to, for example, aphids and so on, or other pests sometimes. Mealybugs, for example, they can be very. But yes, there are some parasitoids, especially of braconids.
Excellent. Here's another question. At which time, like after how many months or years after the detection of the fall armyworm in a new country or region, do you make the choice between the three concepts of biocontrol?
Yeah, I don't think there is necessarily a choice to do. For example, with fall armyworm, we are doing the three in parallel. At CABI in our fall armyworm program, as we said earlier, we have a classical biocontrol project, but we are also developing methods for rearing and producing Telenomus in Africa and Asia, and our colleagues in Africa are also developing methods to conserve natural enemies. They are not mutually exclusive, and especially conservation and needs to be done even when you do a classical biocontrol. You usually need to conserve the parasitoids or the predators which have been introduced as well as the native ones. So they are not mutually exclusive at all. So and the sooner the better.
The sooner the better. Excellent. Okay, thank you very much. What we might do, Marc, is I know you've got a lot of information to come, and we're getting lots of questions in. What I might do is move forward and get into the sort of this big meaty second section of your presentation, and then we'll come back to questions. Okay. So I'll move. Yeah. I'll move on to the next slide, and you can start.
Okay. Then here is about assessing the natural enemy complex and their impact on fall armyworm, how to do it. Well, there are different levels of difficulty, whether it is about studying parasitoids and predators. These are very different techniques to be used. And also whether you want qualitative assessment or quantitative assessment. It's very easy to have a qualitative study of parasitoids, so just a list of parasitoids attacking different development stage with an approximate and relative level of abundance. It's a bit more complicated with predators because you don't necessarily sample them in the same way. There are different predators which you sample in very different ways, but it's still possible to have a qualitative list of the predators present. It becomes more complicated when you want a true and correct quantitative assessment of the natural enemies, which means what is the role of these parasitoids and predators in the mortality of the fall armyworm. It's difficult with parasitoids. It's more difficult than you would think to get a true parasitism rates, and it's extremely difficult to get a true assessment of predator rates and the role of predators in controlling the pest. There are not more than 2 or 3 studies on predators in the whole history of studies of natural enemies of fall armyworm that somehow try to assess the role of predators. There are much more parasitism rates occurring in the literature, but most of them are not properly assessed. Next slide. So we start first with parasitism, because I said parasitism and predation are 2 very different assessments. So it's not easy to assess them together, at least not with the same methods. I will start with qualitative assessment, how to get a list of parasitoids attacking fall armyworm in your region with an approximative idea of the respective roles. Well, this is very easy. For the egg parasitoids, you search for egg masses on leaves, and so the egg masses are quite visible. You collect the entire egg mass. You may note if they are apparently parasitized. The parasitized ones tend to be darker in color, but also the ones which are unparasitized but are ready to hatch are also dark. So it's not that easy, but okay. So when you collect them, you need to place them in a cooler during transport, because in a car you quickly get very high temperatures, and then especially when you don't have a good air conditioning, then it quickly dies. So you have to put them in a cooler during transport. In the laboratory, you have to place the egg masses singly in a closed tube or closed petri dish, very close, because these egg parasitoids are very small. You need to keep the humidity above 50% in your lab. So depending on the season, it may be more or less difficult, because these eggs, and also the same for the larvae, they dry out very quickly. You have to avoid direct light to avoid a greenhouse effect in the things. This is kind of regular procedures for collecting insect, of course. This is nothing to do especially with fall armyworm. You have to know the emergence of the larvae and the parasitoids and the number, and you have to place them in alcohol, or if you want to rear them, you can rear them. Next slide. For the larval parasitoids, well, it's a bit the same. You have to search for larvae on leaves. The first thing, of course, is to collect the right larvae. It's more difficult than it seems, especially when it's a new species in a new environment. You have to use identification guides. If you are not familiar, depending on the region, it may be more or less easy, depending on which native species also occur on maize. So there is no special guide here. You really have to use a guide for your region. So the larvae are cannibalistic. This is very important. You have to place them separately in tubes or in small box or petri dishes with a piece of leaf, especially leaves which have not been sprayed. Be careful with that. Or in small, if the transport is short, you can also put them in small groups in bigger container with full of leaves, where they are much more likely to encounter leaves than larvae, because they are quite aggressive. And especially the problem is that the ones which are not parasitized are much more hungry than those which are parasitized. So if you leave them together, at the end it's only the unparasitized ones which will remain in the container. You also have to place them in a cooler during transport, very important, otherwise you may get them all dead when you arrive in the lab. Yes, next slide, please. You have to rear them, all the larvae, separately, as I said, because of cannibalism, either on diet, which you can change depending on the diet, depending on the condition, every 4 to 7 days. Sometimes some diets can last longer, it depends. Or on leaves. If you use leaves, of course you have to use untreated leaves, for example maize leaves that have been grown on station that you know that they have not been sprayed. You have to change them every 2 days, otherwise it dries too quickly, especially if the relative humidity is low. What is very important is that some parasitoids require substrate to make cocoons, especially braconids and ichneumonids. They make cocoons in which they pupate, and with several of these species, including the most abundant ones, they need some kind of substrate to make the cocoons. Otherwise, it does not work. They never make cocoons, and the larvae die, because if there is no cocoon, the larvae can start making a pupa, but it will never become an adult. So you can, of course, what a possibility is to leave when you are close to developing the parasitoid, you may leave the old maize larvae leaves in the petri dish and put new ones in case they want to feed more. But if you use them, if you use diet, or even if you use leaves, a good trick is to put cellulose paper, like toilet paper, for example, at the bottom of the boxes or in the tubes, so that this will allow the parasitoid larva to make a cocoon. You can see there on the below, on the lower photo, you can see cocoons of Kenis species, which without this paper would not be able to make their cocoons. This is in group because it's a mass rearing. It's not individual rearing for parasitism, but it's the same principle. When cocoons or pupae are obtained, you have to wait for the emergence of the parasite. Oh, sorry, not yet. So when cocoons or pupae are obtained, wait for the emergence of parasitoids. Keep the cocoon with the parasitoid. It's very important when you want to make when you want to identify them. It's always good to keep the cocoons with the parasitoid, especially when this is a rare parasitoid. It can be actually a hyperparasitoid, and using this method, you know whether there's something totally different comes from a cocoon which is known from a primary parasitoid. You can put in alcohol or in the freezer for preparing insect. I will mention this in more detail later on. Next slide. So of course, in most cases, people are doing that. This is not rocket science, which I mentioned. This is just regular procedures for many other Lepidopteran parasitoids. The study of their parasitism. But with these data, lots of people are also mentioning percent parasitism, which is usually calculated, for example, by dividing the number of parasitized egg masses by the number of collected egg masses, or the number of larval parasitoids obtained divided by the number of collected larvae, or a little bit better, divided by the number of larvae successfully reared to pupa or parasitoids. Next slide. This is wrong. This is with this kind of method, you won't get a real assessment of parasitism. And the reason, there are many reasons for that, which I can explain in a few words on the next slides. What are the issues? So first, starting with egg parasitoids, what are the issues with egg parasitism? Well, first, the parasitized egg masses stay much longer in the field than unparasitized egg masses. It can be 4 times longer for Telenomus remus. So it means that in a normal tropical weather, the fall armyworm egg masses will hatch after 2 or 3 days, and the Telenomus remus, the ones which are parasitized by Telenomus remus, the parasitoid will come out after 10 days, 12 days. What does it mean? It means that when you will sample eggs, you will find much more frequently parasitized eggs than unparasitized eggs. And also the parasitized eggs stay a long time because they are quite dark and they are quite visible. So if you just survey for egg parasitism, you will largely overestimate egg parasitism if you don't take this into account. Next slide. And the solution, of course, is to study parasitism in a field where the same plant can be inspected every day. Like, for example, if you work on station, you can go every day to inspect the same plants, and you can tag the egg masses with the day of finding to be able to follow all the egg masses. And then you can follow all the egg masses day by day, and then you can have a much better idea of which percentage of the egg masses will be parasitized or not. Of course, in this case, you will also have some egg masses disappeared by predation and so on. It's not ideal, but at least it will resolve at least a part of the bias. You can also use sentinel masses if the egg masses are too complicated to find, because for example populations are too low. You can put egg masses which have been laid in the lab on paper on maize. This is not the exact natural situation, so you may have a biased parasitism rate idea as well, because lots of parasitoids are especially attracted by the pheromones by the pheromones of the moss and the plant, and so they may not. It may not be an exact idea, but it can greatly facilitate the assessment of egg parasitism, and this needs to be very fresh, because lots of these parasitoids, they lay eggs only in the fresh egg masses. Also, don't forget that not all eggs of egg masses are parasitized. You have to measure this parameter also in the lab. That was for egg parasitism. Now for larval parasitism, what are the issues? Well, an important issue is that early larval parasitoids will kill their host before larval maturity. For example, if there are 16 star, they will kill their host in the 14th star, sometimes in the 15th star, but not in the 16th star in most cases, for most larval parasitoids. And late larval sorry, it's not yet the solution. And most larval parasitoids will attack large larvae. Like, for example, the tachinids, several tachinid species will only attack the mature larvae. Thus, if you collect larvae of all ages, you will underestimate larval parasitism. I just can give an example. Oh, sorry, yeah, next slide, please. And the solution is to collect different larval stages, not only the big one, that would be a mistake, but all the larval stages, especially from the 3rd instar on. Note the larval stages at collection and at death from parasitism. That's very important as well. You need to know which instars you collect and which instar are killed by this or that parasitoid. You have to use the head capsule width, not the body size, to know the host instar. This is a very well-known thing, but think of that, because lots of people still use the body size to assess the host stage, but this is not really correct. There are publications with the head capsule width, several publications on that. And then you have to add and integrate parasitism at the different larval stage to calculate parasitism. If enough data are available from different instars, a partial life table calculation can be used. I can just mention in the next, show in the next slide, please. A very simple example. Usually the cases are much more complicated than that, but let's take a very simple example to show what a partial life table approach means. Like, for example, someone's collect 200 larvae. Half of them are small, medium, and half of them are mature larvae, last instar. In both categories, you have 20% parasitism. one by a braconid and the big larvae by tachinid. So most people, or the natural reflex will be to say, well, parasitism on average is 20%. But this is, of course, totally untrue. If you are looking, for example, so a life table approach would show that if you start with 100 larvae, this is theoretical, if 20% die in the first 4 or 5 instars by one parasitoid, you have 20 dead, 80% survive. Among these 80% which survive, 20% are parasitized by the tachinids, which means 16 dead, which means that 36% of the 100 larvae will die from parasitism. So parasitism in this case is 36%. It's not 20%. And I think that's very important. And these are very important errors that are done in many studies on parasitism. They underestimate parasitism as much as they overestimate ecparasitism. So next slide. Then another issue which I found out when I started working on that in Latin America to survey for parasitoids there, is that is the fact that parasitized larvae are much less visible. They do much less damage, and thus they are often much less collected than unparasitized one. If you see at the graph, this is a graph that was done by my colleague Koku Agboyi in Ghana by when studying one of these small instar larvae parasitoid, Coccygidium luteum. You can see that the unparasitized larvae eat far more than the parasitized larvae. And you can see here, so on the bottom image, the two larvae, unparasitized and parasitized, at the same instar, just before or at least at the same age, just before the parasitoid will kill the parasitized one. So what does it mean? It means that the damage and the size of the unparasitized larvae in the field is far more visible than the parasitized one. And so, especially when people go to farmers' fields, they want to collect a larvae, but they will go immediately to the plants which are heavily attacked by fall armyworm to make their samples, especially when the farmer sits beside. You don't want to dig out an apparently healthy plant, but it's usually in the apparently healthy plants that you will find the parasitized one. So next slide, please. So to overcome that, you need to search for larvae in plants with low damage as well. Not preferably, but just as well. And this means that you have to do destructive sampling. For example, you can do that on station rather than at the farmers who doesn't like you to destroy his healthy plants. And this is very important. You have to select your plants before and then check every single small parasitized larvae which is not visible from outside. Otherwise, you will again, this is another reason to underestimate larval parasitism. And this is also partly why in the literature in the Americas, you have usually very low rates of parasitism in the literature, which especially larval parasitism, which is caused by these two main issues. Next slide. Then a few additional advices to assess parasitism. Of course, you have to know the factors that may affect parasitism. Plant growth stage. There are various scales available in the literature about maize or other plants growing stage. The level of fall armyworm attack, there are also various scales available, or you can make your own scale. Management methods, whether it is sprayed or not, whether there is intercropping or not, and so on and so on. So also for the reason I mentioned, but I repeat it, you have to do randomly selected plants for quantitative assessment. And the field sampling protocol can be followed, for example, using a zigzag or W scheme, like, you know, just, but of course you can do your own field sampling protocol. You don't necessarily have to follow protocols which are used by others, depending on what you have available. Note all the regular survey data, like the coordinates, the dates, the collector, and things like this. And as I said earlier, develop your own sampling protocol depending on the local situation, infestation, funding, objectives. The objective is important because it may decide whether it is better to make frequent samples at the same sites or to replicate sites with 1 or 2 sampling only. It depends on the objectives. That's why I don't like to give very precise protocols, because the objectives can be very different depending on which factor you want to study. Next slide. Okay, that was for parasitism. Then predation is the same. Qualitative assessment is easier than quantitative. But in this case, there are many different types of predations and predators. So of course, there are many different collection methods. If you want to study parasitoids, you have to collect the host. But if you want to study predation, you can't really collect the prey, because the predators are not necessarily present when you do that. You can of course observe direct field observation. A field observation of predation, like a predator actively feeding. That's of course, that's very useful, but it's, unless you stay long in the field, it's not, you don't often get that. You can use visual counts of supposedly predators on randomly selected plants. So you select, as for parasitoids, a certain number of plants using a certain protocol. And then you check the plants very well, and then you check whether there are ants, whether there are earwigs, and so on the plant, or bugs, and so on. And then you collect the predators. And of course, what is very important, if you haven't seen the predation event on the field, you have to test the predator in the lab by offering eggs and larvae to make sure that these are real predators of the fall armyworm, especially in a newly invaded regions where the predators have never encountered the armyworm. Next slide. And quantitative assessment, this is really a problem. Assessing predation rate, which is the mortality due to predation, is very complicated. Calculation implies a life table approach, as I showed simply for parasitoids, which means assessing mortality rate at different stages of development. But it's more complicated for predators than for parasitoids, because when you do that, you just know that the prey has disappeared, but you don't know by what, for example. So it requires a good idea of the main predators first in qualitative assessment in advance to develop quantitative assessment methods. It's even more important than for parasitoids. It can only be done at a later stage, after you know very well the biology and the efficacy of the most important predators. Potential techniques include sentinel eggs, or larvae and pupae. So by exposing material from lab rearing, the sentinel eggs and pupae are quite easy to expose. The larvae, that's why I put them in parentheses, it's much more complicated, because the larvae are moving a lot, and they often disappear, so it's much more complicated. You can also use exclusion cages. This means that putting cages around some plants and not around others, to see the difference between the protected plants and the ones which are not protected. Or you can protect them also by banding the the stems and things like that. So there are different methods. The fact is that by, like, for example, by using exclusion cages, you don't necessarily know which predator will be prevented from attack. And you can also use, of course, with more sophisticated things, cameras, for example. That's it's also complicated because lots of these things happen at night. So the fall armyworm is partially nocturnal, and so they are often during the, when the sun is shining, they are hidden in the plant. So it's not so easy to study that. Next slide, please. An intermediate solution, it's quantitative data on predators' abundance, and this is what usually people have done. Few people, but some people have done when they are studying predation, to have at least quantitative data, which means that when the main predators are known, for example, it's mainly an earwig or a bug, their abundance can be measured to assess the influence of various factors. For example, for conservation biocontrol. Like, for example, you can measure their abundance in intercropping versus monoculture. You can measure their abundance in a pesticide treated plot versus untreated plot, and so on. And then, even without knowing the exact predation rate, at least you know which cultural practices will favor predators of which that is an important predator of the fall armyworm, even if you can't quantify it perfectly. Different trapping and counting systems can be used depending on the predators. You can use bait traps, for example, for ants, or you can use pitfall traps for carabs and others, sticky traps for flying insects, visual counts, sweep nets, etc., etc. But this really depends on the main predators which you want to study. Next slide, please. Okay. I think I finished the second part. Alison, if you have questions.
Yeah, thank you so much, Marc. That was incredibly interesting. I was really struck also just by those tips as well around making sure you don't underestimate the parasitism. I thought that's very useful, and I know that talking to some people on the chat here, there's a lot of people doing work in this area, and I know that sort of advice is going to be very helpful. I have quite a few questions here that would be good to ask you, noting that we've just got to go a little bit faster in the next section. So here's a question here, Marc. Is it possible to get natural enemies if we keep 1 or 2 rows of crops in the farm unsprayed while the rest are continued to be sprayed?
Well, it depends on the natural enemies. It depends, of course, it can be helpful. But it needs to be studied case by case. I'm not aware of these techniques on fall armyworm with the fall armyworm natural enemies. It may be helpful with some, I guess with species which are moving a bit less or more. So it may influence the success of this technique, but I'm not aware that someone has tried this technique for fall armyworm and its natural enemies.
Okay. Here's another question from David. What type of diet do you feed the fall armyworm larvae in the lab?
Well, in Switzerland, because the manpower is so expensive, we buy the diet, already made diets. I mean, there are lots of things like like wheat germ, like CABI. I mean, there are recipes everywhere in the literature. I don't exactly remember what all the components are. Of course, in countries where manpower is not as expensive, it's much better to do it by yourself. But I mean, the recipes are in the literature. There are various recipes are usually based on wheat bran or wheat germ and these kind of things with proteins, with vitamins, with antifungic components, and so on and so on.
Okay, here's a question here. Is it possible to find references of identifying instars by using their head capsules?
Yes, yes, sure. I can send these. This is this is available in many literature. I think there is even a publication that has reviewed all the different measurements, because the measurements can vary from one study to the other, because depending on the food you give them, you may get, especially in lab rearing, you may get very different results. You may even get much more instars than 6 instar if the food is not ideal. But so that's why it's always better at the beginning, when you have your own system, to try yourself first in the region, in your system, to try to measure this first. But the most of the field data are quite similar. Okay. So yes, they are available. I can provide this literature.
Excellent, thanks, Marc. Here's a question here. If our candidate biocontrol agent is nocturnal, what other parameters can we look at in the field other than field abundance to relate it to the fall armyworm population to say that it is a potential agent? Of course, aside from the assay tests we usually do in the lab.
Yeah, well, I mean, usually the nocturnal natural enemies are rather so the predators. Some predators are more active nocturnally, and of course, in this case, you have to use different traps depending on the predators. You have to use, for example, pitfall traps, sticky traps, and so on, which will also catch the nocturnal predators.
Excellent. Okay.
Yeah, this is a good thing that I didn't think of it, but I'm wondering about nocturnal parasitoids, whether they also occur nocturnally. This is just reminds me that a question that I haven't thought about.
Well, that's good then, Marc. I think that's a good outcome to give you more to think about, because we need all your mind power on this topic at the moment. Here's a question here, and I guess it gets back to this pesticides in the field, where if we release trichogamma in the field, how can we know how much its release works, because we cannot manage farmers not to use pesticides in the field?
Well, I think using pesticides with augmentative biocontrol is very complicated. Yeah. It's at least at first, all the trials have to be made without pesticides, and then case by case, you can see whether you can integrate. But usually, pesticide in integrated pest management have to be used very cautiously, and only when it's deemed necessary. And in most cases, when you are using augmentative biocontrol, it is to avoid the use of pesticides.
Okay, great answer. Okay, we're going to move on, Marc, because we don't have too much more time. We're at 5:09 in my time, and we're going to finish at past today.
Yeah, the last part is quite short now.
Yeah,
I mean, but this was not well equilibrated between the two.
No, that's fine, because we can go through that, and then we'll leave the last bit for questions. So if you have questions, here's your chance to really put them in the Q&A box and get them answered at the end of this session. So please do, because you've got a real expert here. And also your questions will sort of be collated along with the chat, and we'll follow up through this whole 6-month series. So it's a really good chance for you to sort of get involved and get interactive. So please use the Q&A box. And Marc, I'll just move to the next slide for you now.
Yes. Okay, so identification of natural enemies. Once you have collected them, you need to know what they are. First, a few general remarks. All adult natural enemies obtained from rearing or survey material should be kept, because there is sometimes the tendency to keep just a few specimens and to throw away the rest because you think it's the same. But you have, of course, the possibility of closely related species. And then when you realize that there are two closely related species, it's too late, because you have thrown away most of the specimens. So it's important to keep every specimens. Not all, you have to expect that not all the natural enemies will have a name, especially in a newly introduced region. You may collect a lot of parasitoids or predators that are simply not described, or will have be given a name, but which will finally become an unknown species. That's quite common as well. Sometimes people tend to give a name, but at the end we realize that this is a species that has never been described. So you don't expect all natural enemies to give a name. You have to work with specialists at national or international level. It's always important. Don't try necessarily, well, of course you can try. You can try to try yourself first to separate the different morphospecies. So specimens that look like the same species, but you need interaction with the specialist to improve your knowledge at the beginning. It can be a taxonomist or molecular biologist or both, if preferably, but it has to improve. You have to work with that. Don't try to publish, for example, without at least showing your specimen to a specialist, unless you are a specialist yourself. Yes, next slide, please. So you have two different main method to identify natural enemies. Through the traditional morphological identification by taxonomists, specialists in their field, based on identification keys and collection specimens in museums and so on. Or barcoding by molecular biologists, usually the one, so for insects, it's the CO1 gene which is used, which is called the barcode. There are more and more insect species which are barcoded in the literature or in the banks, in the barcoding banks. But you have to expect that many parasitoids are not. It's even much better than 3 or 4 years ago, but it improves a lot. But you still have this problem, and you don't have to expect that all the specimens which have a barcode in the barcoding systems are correctly identified. That's another issue, of course. Next slide. But what is very, very important to keep in mind is that these two techniques are complementary. They work best when used together. Don't rely necessarily only on barcoding to have your insects identified. It's always good to have the morphological backup and to make sure that these identifications make sense and so on, because sometimes the barcoding end up with no sense results. So it's important to work with taxonomists which know about the morphology of the species. That's very very important. I would insist on that. Next slide. So about preparation and conservation, for the morphology, specimens are killed alive, should be killed alive, because they are easier to prepare. If you take dry insects at the bottom of your cage or your container and then try to prepare them, or they will immediately break. Of course, there are techniques to make them softer and so on and things like this. But it's always better when you keep them as soon as possible alive, or when they are very freshly dead. Freezer is, I found putting them in the freezer the easiest. It's not necessarily the methods where they are the best presented, but still it's by far the easiest and the least toxic. There are some other toxic components that are used to keep them well softened, but that's not honestly, I really prefer the freezer myself. Or you can also put them in alcohol at 70% if you think that they need morphological preparation for morphological identification. If you put alcohol with higher percentage, they become extremely breakable. So it's not good. On the other hand, for the barcoding, the best is to put them in alcohol at the highest percentage possible. I mean, the people who are, molecular biologists will tell you to take the highest alcohol percentage as possible and put them in the freezer. In reality, they can still usually, in most cases, it can be also analyzed specimens in alcohol without freezer, or in alcohol at 70% with freezer and things like this. There are now, of course, lots of techniques that can also extract DNA with dead specimen, dry specimen. But this is much more complicated. So don't make the life of the molecular biologist difficult. And so if you send them the insect killed by putting them in a high percentage alcohol, that's the best. Next slide. Where and how to send it to national or international taxonomists and molecular biologists. If you have national taxonomists who you trust, that's usually better because you have national collections and so on and things. If you want to use international expertise, you have to follow the access and benefit sharing regulation of your country. Every country has different regulations. There are countries from where it's very easy to send a specimen to another country. There are some countries from where it's extremely difficult because of very strict regulations. So you have to be aware of that. But most people, most entomologists will know that, especially those who work on international issues and so on. I can advise, but I can even receive samples, and I don't do molecular analyze myself, but I can find ways to do it with my CABI colleagues, but only if it's parasitoids and only if it's from fall armyworm. Please don't send me any kind of natural enemy, also from other pests, because I will not be able to handle that. But of course, with now our expertise on fall armyworm parasitoid, we get more and more samples from everywhere. We have the corresponding barcodes and so on. So it becomes quite easy for us to identify or to advise where to send them. Sorting to morphospecies by the scientists themselves or the taxonomy group is important because first, because no specialist covers all group. You can't find any specialist which can identify to species level tachinids and braconids, for example. Even sometimes there are specialists, world specialists that only on one or two subfamilies of braconids. Then of course you have more generalist one. But even there, they are not able to identify to the species level all specimens. So it's good to sort to morphospecies, but also for you, also for the scientists, it's good to send to morphospecies, because then you can at first send only a part of the specimens or of the abundant species. The rare species you may want to send it, but if you have a lot of the same specimens having the same cocoon, exactly the same look, then you can first start by sending the most different of all these specimens. If possible, keep voucher specimens and make a reference collection. That's very important. Provide as many details on the sample, whether it is for a molecular or morphological identification, the host plan, the date, location, collector. If from cocoon, it's good to provide the cocoon as well, especially for morphological identification. If barcoding, it's usually now the airplane companies, the flight companies ask to remove the alcohol before shipment and ship by, and then of course then you have to ship by express courier so that people who receive them can fill them in when it arrives. If it's within a week, there is no problem. You can easily do that. You can also ask the molecular entomologist, the molecular biologist, sorry, to use non-destructive methods. In the past, the insects were squashed, and you could not identify them later. But for big insects, of course, you can use a leg, or even an antenna or things like this. But now there are non-destructive methods that can be used so that the insects is dipped into the, you know, the products, and then you still get your specimen for control after that. And then a last note: for quantitative studies of parasitoids, it is important to identify the parasitoid larvae that fail to develop to the adult stage. Sometimes it is very abundant. It is very frequent in some studies that a lot of these larvae can't go to the adult stage. If identification is not possible through morphological characters from the larvae, you have to keep dying of freshly dead larvae or pupae in alcohol for barcoding. That's also possible, of course. Next slide. I think it's, yes, and last advice. I would strongly advise you to collaborate. Don't be afraid to share experience and results. Science is much more fun when you share your results, when you work together, when you publish together, when you discuss, when you exchange samples and so on. This can be done through various ways. At CABI, we have a CABI fall armyworm research collaboration portals. For those who are not yet there, they can go and they can inscribe themselves and chat with or ask questions or describe their studies or upload the natural enemies found in the country and so on. We also have a newly made IOBC, International Organization of Biocontrol subgroup on fall armyworm biocontrol. We are going to organize meetings, especially after the COVID crisis, but even now we are going to try to organize an online discussion. Of course, this is more for scientists really working on biocontrol. And there are plenty of other opportunities to share and collaborate. So so have fun when and we keep contact.
Great. Thank you so much, Marc. And I definitely think having fun is always a good recommendation, particularly combined with any science endeavour. So I've got a couple of questions for you. We haven't got too long for this, but I do want to get through as many as possible. So here is one. Do you use ethyl acetate for killing your predators?
Yeah, you can. I mean, the insects are very well spread when you kill ethyl acetate. I don't like to use this chemical myself for my own health. I'm not too sure what it is the exact status in terms of toxicity of ethyl acetate these days. These things change depending on what the. But I've stopped using ethyl acetate 20 years ago when I got a freezer in my lab. It's just easier. But I agree that the insects are kept more spread and are easier to prepare when they are killed by ethyl acetate.
Okay, thanks. Here's another question. Which are primer pairs specific for identification of the Kenis species and the Telenomus species in the molecular method?
Well, I mean, these are very easily identified through the simple CO1 barcode. So we don't need any others to get the identification with that.
Okay. Another question. Are there any possible pheromone or volatile compounds to enhance the attraction of parasitoid to fall armyworm?
Yeah, that's a good question. I don't know. I don't think so. I don't think that, but maybe I'm wrong. I've never looked into that, but it could only be with egg parasitoids then, of course, because of course the pheromones are emitted by the females, but not necessarily when they lay the eggs. So I don't know. I don't think that it is a good way to enhance. I mean, it depends. There are some insects which don't move very much. Like, I mean, thing which come directly into my mind are mealybugs, for example, scale insects, where females emit pheromones to attract winged males. They are themselves not winged, but the males are winged, so they come, they mate, and then the females lay their eggs. So they can, then parasitoids can use this to locate females and their progenies. With the fall armyworm, it's possible that that the egg parasitoids are attracted. I'm sure they are attracted by compounds that are used, that are included in the egg masses. These are not directly the sex pheromones, I guess, but maybe I'm wrong. I don't know.
Okay. Well, the person who asked that question might have some more information. Maybe they're looking at that. So please contact us. But we'll be sending out an email to everyone anyway, so that'll be a good chance to sort of respond to us as well. And we'll pass any questions or any of the research you're doing on. I've got another question here. In your studies, Marc, did you see more or less parasitism in relation with the size of the field? I'm thinking of commercial fields for seed production, as in some places in Kenya.
Well, yeah, for the moment, we are still studying that. I mean, these studies take time, and the invasion of fall armyworm is recent. So I don't know. I don't know for the moment whether there are. I mean, I know that some of our colleagues are presently studying the effect of monoculture and versus intercropping, but usually they are using small plots, not very large plots. So I don't think this is known. Maybe some people in Kenya, like Isipe, have already studied that, but I don't remember seeing that in the literature.
Great, thanks, Marc, and thanks for the question, Pierre. And last question here: as Telenomus and Trichogramma can attack fall armyworm, is there a possibility that this multi-parasitism leads to the death of one of the parasitoids?
Yes, for sure. I mean, usually in an egg, you cannot have I don't think you can have a Telenomus and a Trichogramma coming together. But on the other hand, Trichogramma and Telenomus have been tested together by different scientists in Latin America, but also I think in Africa, and the results are quite positive. So you can combine the two, but of course, per egg, you will not have a Telenomus coming out with a Trichogramma.
Great. Well, we're going to end it there. And I'd just like to thank you very much, Marc. I mean, that was a great session, really good practical tips. And it's wonderful to have you share that with us. So thank you very much from all of us. And we'll be in touch anyway. Marc will be continuing to work on this with us. And so any of your questions that come through from the email that I send out, or if you just want to email me or Marc, you're most welcome to, and we can follow up. So remember, it's a 6-month sort of series, so we really want to keep that conversation going and connect all the different laboratories and all the different sort of field efforts that you're doing out there as well. And I noticed a lot on the chat, so it's very good to see. Thank you, Marc.
Thank you for the invitation.
You're most welcome. And I'm just going to introduce Roger. Roger always gets the sort of very sort of squeezed section at the end for the summary. I normally tell him sort of 5 minutes, but then make it 30 seconds. But he is wonderful at doing that on the spot. Roger, welcome back.
Thanks, Alison, and thanks a lot to Marc for that highly informative presentation. Just to emphasize Marc's point about collaboration, Alison will be, I know, setting up ways for collaboration within ASEAN, but also this is a global community of researchers that we can all be part of. So feel free to collaborate in the ways Marc has suggested. And the other thing is that it's very interesting, this research, and we can get really buried in the details, and that's we need to, but let's also keep in mind the farmers who are eventually trying to provide solutions for. It's no good doing lots of very good research if we don't in the end help the farmers solve this huge problem. So keep that in your mind, even as you look down your microscopes at the small parasitoids we're talking about. Thank you, Alison.
Thank you very much. I'll just put my video on at the end too, just to say hello and farewell to everyone. So thank you so much. And it's a very good point to end on, to remember what we really have to achieve on the ground is control of fall armyworm, and that we have to partner with farmers to achieve that. Just a quick shout out again. I know I've mentioned it. Please join us for the rest of the series. It's open participation. Tell your colleagues who may be interested, or your students, to take part in one of these 8 sessions. And of course, we'll have a big sort of workshop. This is the aim at the end in June. And hopefully, you know, some of that could be in person, but we never know, it could be online as well with COVID. But thank you so much for joining us, and we hope to see you at the next session in early January, and stay safe, and we look forward to your participation and your interaction going forward. Thank you very much. Thank you, Roger. Thank you, Mark. Thank you, Graham, and Pranav. Bye.
Bye, everyone.